All right, and welcome to the second week of class. Great job getting your course prep check-in and those first introductions. If you don't see a grade for that, let me know. That means I probably didn't see it, so I comment on them. So hopefully you've seen that. So this next week, we're really jumping in to Mod 1. Hopefully you did number 1. I'm going to do sections 2 and 3 of it. Got a quiz due at the end of the week, and the blog about child happiness that I see a lot of you writing, but really just talking about what do we think about happiness and, what is a great hope for a child or a student. So we're going to jump right in to reading of chapter 2. I'm just going to summarize the slides. Hopefully you're reading along, but if not, this will be a quick little summary for you. So this chapter is all about scientific method, which is basically a huge component of research methods. So we want to formulate a hypothesis based on a theory. We want to make sure that we can replicate these techniques, collect, study, analyze, things like that. And so, because just if I do it once, it's not really scientific method. It was just fortunate that it happened. So we want to make sure that other people are able to do this as well. Okay. One piece of this is the correlational research method. You might remember this from an intro psych class. We can range from +1 all the way to -1. Really, the closer to 1, either positively or negatively, the stronger the relation. And the closer to 0, the weaker. So a positive 0.99 is a really strong relationship, just as a negative 0.99 is also strong. With correlational research, we can't always design a suitable experiment to study our question. So some variables of interest really just can't be studied in a controlled way. There can also be some ethical concerns with correlational research. We can't randomly assign families to abusive or non-abusive groups. Okay, another type of research method we have is experimental method. So this is how we really study causal connections. In an experimental group, researchers assign each participant to a group. So we have our experimental and our control group. Our experimental group is the people who are getting our causal factor, while our control group is our participants who really aren't exposed to that factor. In experimental, we really want to use random assignment because it rules out the possibility that people in different groups can differ from one another in some way that could really distort our experiment. Experimental method is where we have an independent and a dependent variable. Our independent variable is the factor that we as the researcher will deliberately manipulate in an experiment, while our dependent variable is what we're expecting to change. So my independent variable might be if I was giving a group sugar versus giving a different group no sugar, and my dependent that I'm studying is, will their energy level change? So remember that independent is the thing that the researcher controls. We also have cross-sectional groups that we can study, and that's where we compare groups of individuals of different ages at approximately the same point in time. So I'm going to study 15-year-olds, 30-year-olds, and 45-year-olds all at the same period of time doing the same experiment. This is different than a longitudinal study, where we're studying the same individuals over time. So I have my group of 15-year-olds. 10 years later, I'm studying them at 25. Another 10 years later, I'm studying them at 35. So just like with cross-sectional, there's pros and cons. If we're studying over time, we can have a practice effect happen. It's the same people. They know what to expect. It's pretty difficult to do this. And also, people who are surviving every 10 years might change how our experiment is versus the people who didn't make it for whatever reason. And so both of these are great ideas and research approaches, but we want to make sure we're understanding the pros and cons. Let's talk a little about data collection. A couple different ways to do this. We have self-report. This is like surveys and things like that. It's the person you're giving the experiment to fills it out about themselves, a questionnaire. Great to gather a ton of data, but our cons can be people aren't always truthful. They think about, I wonder who's going to read this, so I'll change it to how they want it to be answered. So self-report, again, pros and cons. Naturalistic observation, you probably remember this from intro psych. Basically, we're just observing. We're not changing anything about the environment. You just go to a Starbucks and you're taking notes on who orders what. That's naturalistic observation. We have no control over really any of the factors in our experiment. We can also do a structured observation, which is like coming into a lab and then we're controlling everything. Again, pros and cons. So that is it for this second piece of Mod 1. I'm going to go into our next part now. So chapter 3, if you're following along in the book, pages 85 through 112, really talking about biological foundations of social development. We'll talk about hormones, brain waves, DNA, physical appearance, and reflexes and unconditioned responses. So I want to start this chapter talking about how we're biologically prepared for social interactions, from our biological rhythms like our sleep cycles to how we're visually attracted to things that bring out social situations, so like the eyes and the face, to even our auditory skills of preferring high pitch and exaggerated contours. So we're going to talk about a few different theories. First, the evolutionary theory. This is the idea that infants are biologically programmed for social interactions that really ensure their needs are met. So think about babies you've interacted with and things that almost seem like, how did they know that? How did they know to cry to get fed or get held or things like that? So moving on to our brain, we've got our two connected hemispheres. We've got our cerebral cortex, which is this thing that contains cells that control specific functions of our body, like breathing, like moving, like thinking and hearing. Okay, we have a ton of neurons rapidly firing and rapidly forming during our embryonic period of prenatal development. Thinking about that rapid infant growth in our brain, a 2-year-old brain weighs 3/4 the weight of an adult brain. So we've got these growth spurts across childhood and adolescence, and then all these synapses are starting to form. Okay, this starts prenatally, and we have thousands of them by age 3 with a huge spurt as an adolescent. Our brains are programmed to create more neurons and connections than it actually needs, and so that's where this like synaptic pruning starts to happen. Okay, maybe you skateboarded once when you were 6, and you don't really use that anymore, and so we kind of prune it away so that I can make room for speaking Spanish. Two interesting kinds of brain processes. So we have experience-dependent process, which those are really unique to individuals in a particular culture or community. They're unique to your family experience. And then we have experience-expectant process, which these are universal and experienced by humans across evolution. Another topic here is mirror neurons, and these fire when a person acts the same way as someone they're observing. Maybe you've seen someone yawn and then you also yawn, or you're in a sad situation, people around you are crying and so you are also crying. It's kind of like empathy in a way, but when we do an fMRI of the brain, we actually see these parts light up. It's pretty interesting. Last part of this chapter looks at genetics and social development. So how are the things that have been passed on to me by my parents and my ancestors, how do those affect my social development? So we have a large number of chromosome combinations that are possible. We have the 23 chromosomes from a woman and 23 from a man to form this zygote, and there's crossing over that occurs during cell division in the fertilized egg. A lot of different ways we've studied genetics. We look at adoption studies, we look at twin studies, really comparing adopted children to their biological parents or comparing twins to each other, depending on if they're raised in the same household or a different household, and just giving us some more ideas for that topic of, is it nature, is it nurture, or is it a little bit of both? So just to sum up this chapter, we start to see that babies are actually biologically prepared for social interactions. Think about those preferences for human face and human voice. There's also evidence that our brain provides the foundation for social development. Think about those mirror neurons. Think about how our environment contributes to our brain development, whether it's from that synaptic pruning or things that we learn from our culture or things that are just pre-programmed into us. And really the bottom line of this chapter is biology and environment are constantly influenced by each other and work together to shape our social development. So the big surprise is it nature or nurture, or is it a little bit of both? It's really a little bit of both. Thanks for tuning in this long. Hopefully you made it to the end. You are rewarded with the name of today's song, which is See you next time.